Hi, and welcome back. Today we're going to discuss treatments and prevention of cancer. Early detection is still key. Basically, catching cancer before it metastasizes or spreads throughout your body increases your chances of successful remission. This detection can be done through physical exams to detect tumors. Other techniques include biopsy, imaging technologies such as mammograms, blood tests, or DNA tests to search for mutated alleles. And traditional treatments of cancer have been chemotherapy, radiation, and surgery. All of those have problems. For instance, the problem with chemotherapy is that it affects all the cells in your body that are actively dividing. That includes your skin cells, digestive tract cells, and cells of hair follicles. This is not a selective treatment. When cancer patients are undergoing chemotherapy, they lose their hair, they also tend to lose their appetite, and their skin cells aren't allowed to divide. So all these healthy cells are adversely affected. Radiation therapy uses localized radiation to kill cancer cells, and this tends to be used in conjunction with chemotherapy or surgery to remove a tumor. While radiation technology has gotten better to be more localized, radiation will kill cells, and that means it can kill normal cells, or it can damage and mutate normal cells and cause them to in turn become cancerous, leading to secondary tumors, which is sometimes seen in children. There's a newer therapy that uses oncolytic viruses. Onco, again, referring to cancer, and lytic means to lyse or burst. And these oncolytic viruses will specifically attack cancerous cells. Oncolytic viruses exploit special cells of your immune system called macrophages to transport these oncolytic viruses. And the virus will only become active once the macrophages encounter the cancerous tumor. Oncolytic viruses have been used to destroy prostate cancers in mice, and what we found was that as soon as the cancer was detected, the virus began to attack and destroy the cancer's prostate cells. Obviously, the challenge with this approach is to ensure that all your immune cells don't attack the oncolytic virus, and that your immune cells properly deliver the virus to the appropriate cancer target. You wouldn't want healthy cells being destroyed by this oncolytic virus. So remember how we discussed in a previous lecture that steroid hormones, such as estrogen, can increase gene expression? Well, estrogen has been linked to certain types of breast cancer. When estrogen binds to its receptor, this hormone receptor complex causes the expression of genes involved in cell growth and proliferation. Remember the definition of cancer? We talked about that, uncontrolled cell growth. Well, by blocking estrogen's function, certain types of breast cancer can be slowed. We can use certain drugs that specifically interfere with estrogen binding to the estrogen receptor to slow the progression of that type of cancer. The FDA in the United States has approved the use of certain drugs for treatment of estrogen receptor positive breast cancer. These drugs are called selective estrogen receptor modulators, and they destroy that estrogen receptor complex. These new approaches focus mainly on treating cancer cells. A new class of drugs work by blocking growth factors or repairing broken tumor suppressor genes in cancer cells. To do this properly, we have to compare healthy cells to cancer cells and compare the genes that are mutated. Remember that genome approach I talked about earlier, comparing a cancer cell genome to your healthy cell genome? Well, if we know which specific genes are broken, we can use a personalized approach of treatment by classifying the cancer not based on where it originated, like we do now, but based on specific genes that are mutated that led to that cancer. This approach uses the genetics of the specific cancer and specific genes that are broken as the Achilles heel for determining which medications to use to treat that cancer. So in the future, when someone gets cancer, the genome of the cancer can be sequenced and a personalized treatment can be designed that is patient-specific. Doctors can then identify all the broken genes that led to the disruptive signal transduction pathway, and then a doctor can look up in a database and identify the specific drugs that can block the mutated gene products. And using the specific pathways that the cancer is addicted to survive, you can stop the cancer dead in its tracks. Gleevec is an example of how the future will work. The future treatment is to use drugs that are specific for the broken genes and to interfere with these pathways. Of course, cancer cells can mutate to negate even the effect of Gleevec. So you'll need to use other drugs in conjunction with this based on the new mutations that develop. So the idea here is to target specific genes that are broken and just target the cancer cells. So you want to use very specific drugs as opposed to the current approach of blocking all cell divisions. It might take years for this treatment to be mainstream, mainly because the big issue is sequencing these cancer genomes can be very costly. Now I'm going to review some terminology we talked about last time. One was proto-oncogenes, and again, you have proto-oncogenes. They're normal genes that regulate the cell cycle. One example of a proto-oncogene is the RAS gene, and a mutation of the RAS gene or any other proto-oncogene results in an oncogene which will increase cell growth and cell division. The trick to treatment is to find which specific proto-oncogenes are mutated or broken so we can fix them when trying to treat cancer. We can also try to fix broken tumor suppressor genes like P53 when treating cancer. Another challenge of treatment is to locate the cancer cells in a body full of trillions of normal, healthy cells. A cancerous needle in a haystack, if you would. There are factors that increase cancer risk, such as viral infections. How can a virus cause cancer? Well, remember the HeLa cells I discussed earlier? Miss Lax, she had been infected with HPV, human papillomavirus. And that's linked to cancer because viruses bring with them strong promoters. And remember, strong promoters increase gene transcription. That can increase the rate of the cell cycle if the virus causes more growth factors to be synthesized in your cells. You can have inherited cancer genes, but again, that's not the cause of most cancer. A poor lifestyle is the big risk factor. The single biggest preventable cause of cancer is smoking. Smoking directly leads to DNA mutations. Smoking or using nicotine products can mutate our DNA. And anything that can mutate the DNA and increase your chance or risk of developing cancer is called a carcinogen. And smoking is also linked to cancers of the oral cavity, larynx, esophagus, and lungs. Having a high animal fat or red meat diet can increase your risk of colon cancer, and that's because animal fat contains fat-soluble toxins that are carcinogenic. And they can build up when you eat that animal fat and you expose that toxin to your cells. UV can also be a potential mutagen. Exposure to UV can increase your chances of developing a type of skin cancer called melanoma. So I listed some factors that can increase your chances of getting cancer, but it's probably more important that I discuss ways you can decrease your chances of developing cancer. Well, exercise is high on that list. Do you remember why? Exercise increases the activity of your DNA repair system. And that's one big reason amongst the many for you to exercise more. Also, a diet high in fruits and vegetables that are rich in antioxidants, as these antioxidants can help with DNA repair. Avoiding tobacco products like we just mentioned, cigarette smoke, secondhand smoke, since those contribute to mutations of the DNA. Also, avoiding UV radiation by using a high SPF sunscreen. You should also avoid excessive X-rays, which can damage DNA. Lastly, vaccinations for viruses such as the one for HPV. If you prevent your chances of getting that viral infection, you reduce your chances of getting cancers associated with that virus. While there are inherited cancers, lifestyle choices can really help reduce your risk of cancer. We can't control the genes we inherit, but we sure can control how we live our lives. Lifestyle isn't just important when looking at cancer. For instance, type 2 diabetes mellitus has a strong correlation with obesity and abdominal fat. Abdominal fat can act as a hormone-secreting endocrine gland. And by maintaining a healthy weight and lifestyle, even if you have a genetic predisposition to this disease, you can reduce and possibly eliminate your chances of ever having an onset of diabetes. The role lifestyle plays on the onset of diseases is a pretty tricky one to examine. Consider multiple sclerosis or MS, an autoimmune disease that affects your nervous system. We don't understand why in some people their immune system begins to attack itself, but we do know in the case of MS, there's a higher incidence of MS the farther you grow up away from the equator. It may be linked to the synthesis of vitamin D, which is an important steroid hormone that regulates calcium levels. What I want you to take home from today's lecture is the importance that lifestyle decisions have on cancer and other diseases, and that genes don't solely determine your destiny.